Cool, part 2. Almost done, guys. So prophase, you have your x's, and then you want to split them up into like the different like future cells. So when you line them up, and that's called synapsis, the ends, they might be really close to each other, and so they kind of link up, and they may or may not exchange genetic material. Not always, but sometimes. And that's recombination. But then if they don't actually exchange anything, then it's just like normal. You can have errors when you have this crossing over, this recombination, in that you could accidentally take more than you should have taken. You duplicated something or you left something out. And an example of that is called Cree to chat. Sometimes he might ask that question. It's a deletion of part of 5q. So on chromosome 5, you lost the long arm Q.
Can you repeat that? It's a deletion
of 5q, part of 5q. So a translocation is a crossover between non-homologous chromosomes. So like chromosome 1 and chromosome 5. But then if I have 5 and 5, that's normal crossing over. That's normal recombination. But then 1 and 5, like since they're non-homologous, then like that's called a translocation. Single gene disorder. This kind of thing I would throw in the cheat sheet because he'll straight up just ask like, sickle cell anemia has the same kind of genetic mode of inheritance as, and then he would just give you a list of like, all of these, and then you would have to pick out the same like recessive disorder or something like that. So just throw it on your cheat sheet, read them over. With pedigrees, dominant, as long as you have one copy, it's bad. Like achondroplasia, very common cause of dwarfism. Recessive diseases, you need 2 that are bad. Cystic fibrosis, anemia are an example. X inactivation, so because females have 2 copies of the X, you don't actually need both copies. So what happens is all of your cells randomly inactivate one X or the other X. So either the dad's X or the mom's X. And then you get what's called a mosaic. So you have a mosaic in that almost all of your different cells, they're all expressing like either mom or dad. And you kind of have like a nice mix. And that's like what the mosaic is. And so this is important for certain diseases because what if the mom's copy was bad and all of your cells were, you know, inactivating the dad's copy, and so you only express like that mom's copy to be bad, or you know, like something like that. You could inactivate the bad mom's copy and only be expressing the dad's copy, and then you'd be okay. So that's kind of why it's relevant. X-linked diseases. So typically they affect males more because typically you don't see that many X-linked dominant diseases. But X-linked recessive, because it's like one copy is good enough to make a disease manifest. Like a boy only has one X, so that one X, if it happens to be bad, he will have that disorder. So you typically see them more in males. And like sometimes it's good if you guys run through like if a dad has this disorder, if a dad has like the recessive whatever, can he pass it on to his daughter? And then those kind of questions, you just have to run through like, one, is it autosomal? So is it, you know, just a normal like capital T, lowercase t? Or is it sex-linked? Is it like X or Y? And then from there you kind of figure out like, is the dad contributing the Y to the daughter? And then, but he's also contributing the Y to his son. So it's like, will that make a disease or not? And then those are the kinds of questions that like may or may not come up. Down the line, hemophilia, and then last slide, epigenetic modification. So this is like, it basically means like once you have your DNA, you do what's called methylation of certain sites, and this alters what you actually are expressing, like what your genes are actually going to express down the line. And this, it kind of resets every generation. So like, your methylation is set, and then your kids will have their own different set of methylation from you. And if there is like an inactivation of or deletion of the father's side of chromosome 15 long arm Q, then you get a manifestation called Prader-Willi syndrome. But if you get the mom's side deletion, you have Angelman syndrome. And so like Prader-Willi father, Angelman mother, it's just a matter of which 15q you deleted. Did you delete the moms or the dads? And they are very different, like clinically. Like Angelman syndrome, like a lot of those children, they're always like smiling, whereas Prader-Willi, it's kind of like the secondary sex characteristics aren't as defined, or sometimes there's like a little bit of like mental retardation and other things, but they are different diseases depending on which chromosome, like which one you deleted, like mom or dad's. And I think that's it for this.
Angelman, they're always smiling.
Yeah, it's just a,
Some sort of mental.
yeah, it's something like that. Yeah. Anyone else have questions before we end?
This is